It's my pleasure to introduce our next speaker, Dr. Kendall Powell, Senior Director of Bioanalytical at MedPharm. He has a very interesting presentation for us on his company's experience with the QMS. Dr. Powell, it's all yours.
Hi, Sam. Thanks. Can you hear me and see my slides okay?
All is good. Thank you very much.
Fantastic. All that practice paid off. So thanks a lot for thanks for the introduction. And thanks for everyone that's still here watching. You've made it almost to the end. I don't reckon I will run all the way to the full time there, but I'll do my best. So just want to tell everyone kind of a brief story about our company's journey from where we really started as a kind of a pure R&D site to running these bioequivalence IVRT, IVPT studies. Everyone has disclaimers, so I have a couple as well. Just want to make sure if I misstate anything here, that's my mistake, not the mistake of MedPharm Limited. And the second bullet will become clear as we go through the presentation. So also, everyone had a lot of slides going back way back in time. I just had to kind of one up everyone there. So here's a brief history of time. Really, the parts I'm going to focus on really started in 1999. That's when our company MedPharm was founded in the UK. A beginning to end kind of topical contract research organization specializing in semi-solid form dev, manufacturing, and also most importantly, for this series of talks, IVRT and IVPT testing. Fast forward several years there, about 15, 16 years in the future. In 2016 of May, MedPharm opened its first site in the US. It's located here in Durham, North Carolina, where I'm talking to you from right now, with the ultimate aim of making it kind of the center of excellence for IVRT and IVPT testing. 2016 came along and surprised us all with the release of this draft guidance that we're talking a lot about today. And then there's another little gap there that I'm going to talk about. That's going to be kind of the focus of my talk. But in 2019, to skip forward, we actually submitted our first study in 2020. Kind of a little spoiler alert there. We did have a visit from the FDA, and I'll tell you how that went. So if you are interested in the very first part of this timeline, there's a really good book about that. My talk is going to be a little bit shorter in scope and just talk about, how did we get from an R&D site in the beginning of or middle of 2016 to be able to properly run these type of studies under a quality management system that produces accurate, robust data. So I'm going to kind of run through this as we went along throughout the years in this process. So here's a little shot of our lab from literally the first week when we moved in the US in 2016. In May, we had literally 3 people. I was one of those 3 people. John Len, who gave a talk earlier today, has been on several panels, was our, of course, key subject matter expert in all things IVPT and skin related. And we had a biologist as well to do a lot of pharmacodynamic assays. We weren't totally alone. I cannot admit, of course, the UK start of this company, we had over 100 staff in UK for support. And we, of course, leaned on them heavily and continue to work with them extremely closely to this day. But yeah, we had an empty lab at the beginning there. Just in case you have different ways of keeping track of time, a certain movie was released around this time as well. So what were we doing in this first kind of, 6 months of our operation here? Really, we were research and development focused. We were running loads of in vitro IVPT studies with the MedFlux device John Len showed earlier on today in that presentation. We were, in process of transferring, over a decade's worth of IVRT experience from our UK colleagues over to the US, and of course, building other site capabilities and things like that. QMS-wise, we were, again, heavily supported by our UK QA colleagues, completely supported really through all of 2016 because we had no QA staff on site at that point. At that point, also, we were planning for a US QMS based loosely on an ICH Q10, which is pretty flexible, set of regs to basic QMS around. And we were busily writing US specific SOPs. So to just round out the rest of 2016, October, I've shown a picture over here. This is our lab when it's actually finally got some equipment in it. And one of our UK colleagues, Emily over here, instructing us on the right way to do IVRT. And then by September 2016, we had a grand total of 7 folks in the US lab space. Again, still had well over 100 combined between the 2 sites. And then we were all interested to see this draft guidance on Acyclovir come out in December of 2016. So what did we do? Well, we did take some time off for the holidays and New Year. And in 2017, we really kind of started looking at this guidance very closely. There's a screenshot of it to the left. I think many people on this call have probably seen this many, many times. And of course, we read this with a lot of interest, right? This is pretty much up our alley. We certainly support Q1, Q2, and physicochemical characterization studies. That's a little bit out of scope for this discussion here. But what is way in scope is IVRT and IVPT, and those were parts C and D of this intro to this draft guidance. We've been doing it as a company for a very long time. Even outside the company, we had hires within the company that had been doing this for a long time, either at MedPharm or other places, and we knew how to conduct these in the lab. So we did read this and jumped ahead kind of to step C there, right, to see what was up here, and came to this section here, which is suddenly there's a lot of regs and guidances listed, right? So it was immediately pretty apparent that expectations for study conduct for these studies would be high, and rightly so. But obviously this was still a very good fit for our technical skills, so we weren't put off on this. So to kind of update you where we are in time, so it's 2017. The first half of 2017. Really, we're still we're young in the U.S. We're still focused on building out our site and capacity, but we are having some passionate internal discussions on do we want to pursue this in vitro bioequivalence work? Some of the big risks, this is certainly not all inclusive by any means, but some of the big risks is, of course, having a visit from regulatory authorities. As I was prepping for this, I dug through some old emails and found this one down here at the bottom where I'd sent a note to one of my colleagues that just basically says, if the in vitro option as described in the guidance is offered by us, it's pretty much guaranteed the FDA is going to come and visit, and then kind of close it out by saying, look, we really just need to do this the right way, or the risk is extremely high. And some of the other risks, if you're considering doing some of these studies, actually Sam kind of touched on this possibility of study rejection, right? If you're not doing something that is done in a proper way. Also, if you're considering kind of doing these kind of studies, I won't harp on this, but I have some dollar signs down here, right? The red ones are when it costs us money to do something. So there's a financial aspect to this, of course. We're a business. We have to stay in business. And so some of the red dollar signs here, we have to adapt and build a QMS for US site. That's going to be probably more rigorous than we were originally planning. So that's, loads of SOPs, validation, calibration, et cetera. Obviously, we need more staff, right? We're going to need on-site QC, QA, study directors, things like that to manage and properly review these studies. And then once you build all of that, you have to maintain those systems, right? So you have a batch of SOPs and they don't maintain themselves. So that is all kind of considerations that we're thinking about during this time period. And then we got to figure out, of course, how to ultimately, we're a company, so we do have to make some money at the end of all this stuff. We did finally decide, it was going to be in our best interest to pursue this work. If we hadn't decided that, I probably wouldn't be talking here today. But so we did decide to pursue this work, and around the middle of 2017 is when we kind of really started gearing up to, do this work in a way that we thought would be the best possible way to do the work. One of the first steps was hiring a full-time person for quality assurance in the U.S. He was our 15th employee at the U.S. site, and we got kind of a big hitter there. We got somebody with a lot of experience, had been in large pharma for a long time, GXP experience, kind of experienced both roles of GMP and GLP. He really realized the kind of philosophical maybe differences between managing those types of different systems, and he was tapped to really kind of lead the effort here of building that system here in the U.S. So, kind of the first step here that we talked about was really fully assessing the draft guidance on all these, quality aspects, regulatory aspects. If you search through the guidance and you just look for CFR, there's several references, right, to 21 CFR, various parts and sections. There's a reference to 21 CFR Part 58, which is GLPs, and several references to subsections of bioequivalence regs. There's a couple of references to ICH documents, and then there's some guidance for industry documents, right? Some ANDA submissions and more BE testing samples, and then bioanalytical method validation guidance. So what we thought was really key, though, in assessing the Acyclovir guidance, the draft Acyclovir guidance, and all these kind of guidances cited within it, was the context of the wording around those, right? So kind of one of the key ones, and one of the big ones, is around 21 CFR 58. It says applicable principles, right? So what does that mean? We'll talk a little bit more about that, what we thought our interpretation of applicable principles was. And then, again, context around these other ones, maintenance of records and drug samples with respect to bioequivalence testing. We're really just kind of talking about those subsections for those particular activities. I won't go over the analytical guidances, even though I'm an analytical chemist. I'll leave that off for now. And then there was, ICH guidances on GCP, but really just around retention of study records, right? And then, so that was pretty much it, right? But what does applicable principles of GLPs? That was something that, we read and said, well, that's going to require a little bit of interpretation and what to do here. So here's just a kind of thrown together screenshot of kind of the intro to 21 CFR 58, which is GLPs. I'll probably just refer to it as that throughout here. It's cited in the draft guidance, so we thought it best to go ahead and look closely at this. We did have some experience on with these kind of guidances. Our QA expert that we'd hired, I mentioned a little while ago, over 25 years of working on this. Myself, at close to a decade of doing regulated GLP bioanalysis work. So we'd read this a couple of times, but this is reading it in a little bit different way, according to this guidance here. But so, yeah, what do you mean? What does it mean, applicable principles, right? So, I mean, and I know, we just went through it, right? It's not quite this simple. You can't just read the headlines of each of these sections and decide what is and is not applicable. But some clearly aren't. Some of the red lines are drawn through animal care and animal facilities. These are not applicable to what this type of testing, but there are a lot of subsections that are, right? Personnel, testing management, QA, things like that. Some key points that were mentioned in earlier talks, having a protocol. These are all things that are clearly expected, right, of these type of studies, and they just happen to be kind of listed in this guidance as well. So, just a couple of examples out of there. Again, I hope I can be more involved in the discussion this time if we're going to be talking about this specifically. But you need an independent quality assurance unit. I think that's, whether you decide to cite GLPs or not, that's just a good practice for a quality management system. Now, again, these bullets here are out of the GLPs, but do we all agree that these are good things to have, for a well-regulated study? I would say yes. So, maintain a master schedule of studies. QA should come in and inspect the studies. That's something that we think is important. Submit status reports. Really, most importantly, make sure you're not deviating from your own SOPs or not deviating from any cited guidances. Review the final report. Finally, sign an inspection summary, right? So there's a little relatively small section there that you can see over to the right. That's just a screenshot of one of our study reports. So that's the QA phase inspections. They review the study plan. They actually came in the lab and watched the skin integrity testing for this IVPT study and the dosing. They did an audit of the data, and then finally did an audit of the draft report and all the associated dates that they did that. And then finally, a signature at the bottom to show that they've done all those activities. A couple other things about, quality assurance. So GLPs, it's a nice guide for QA responsibilities. Is it enough on its own? Again, I would say no. I'm not in QA, but I've seen them in action. So building a strong QA unit takes more than just what's cited in there. Concepts such as change control for when you're making significant changes to instrument or facilities. Whole process for deviation investigations. If there is a deviation from an SOP, how do you go about that, and how do you document that? correct, kappas. QA loves those, and then you continuous improvement. That's more of a I think a concept that's not necessarily related just to QA, right? But continuous improvement of your processes, both quality and science related. That's just a mindset. I think that's important here. Another kind of key part, right, and this has been touched on in various sessions, is maintenance and calibration of equipment. Again, a couple of headlines from out of the GLPs, inspected, clean, maintained, tested, calibrated, standardized. You should have SOPs to cover your kit. And you have to have records, really most importantly, of all those above activities. Here's some kind of example, standard lab kit down here. What we decided to do, and I think this is fairly common, is kind of break these up into, different categories. How complex is the equipment? What kind of impact is it going to have? Does it even really need an SOP? And then what kind of calibration or qualification do we need to do? And then what kind of documentation do we need? So, what about an extremely simple apparatus like a vortex mixer, right? In most cases, it's not going to have an impact on study data. You might not need an SOP for it. You might not even need to calibrate it beyond what, whatever kind of certificate comes with it originally. And documentation-wise, it's certainly listed in our electronic, equipment log. It has a unique ID, so it can be referenced. But that's kind of the extent of that kind of piece of simple lab equipment. Then you have something that's still relatively simple, but you decide it may have some impact on study data. So you'd want a SOP for that, right? You want to at least calibrate that type of instrument. Kind of a generic example below is a centrifuge, right? You want to make sure it's spinning at the right speed, and if it's a temperature controlled, it can maintain the right temperature, and you'll want that calibration, certificate in your records. And then finally, you have the complex instruments. Definitely, if something's not right with these, it's going to affect your study data in a bad way. You certainly need SOP. These need to be qualified, right? So the concept of install, operational, and performance qualifications are really important for this type of level of instrumentation. And there's usually loads of documentation. There's a little screenshot down there of some of our qualification binders for some of our HPLCs in the lab there. Obviously this whole chat is whole talk conference is about IVPT, IVRT, and qualification of your diffusion cells is obviously critical. That probably falls somewhere in between the last two categories here. If you're using simple diffusion cells, vertical diffusion cells. If you have a more complicated device, if you have an automated device, then that's probably falling into the more complex category, right? You really need to be pretty rigorous about qualifying that kit before you're going to use it on these kinds of studies. Here's kind of a thing, right? Applicable principles. What does it mean? And is this work GLP? I don't know that the answer to that question really matters. I think there's probably some people that have strong opinions one way or the other. In my opinion, the GLPs really provide a nice kind of roadmap or starting point for performing a well-controlled study under a strong quality management system. So that's really the key take home here, right? That's the reason for this. And what are the real reasons? Ensure patient safety, prevent fraud, provide data integrity, allow data to be reconstructed or study to be reconstructed. You've heard that previously several times today. And also ultimately to encourage, different labs to work in a similar fashion. I've heard repeatedly that it's a challenge getting data from different places that are working in very different manners. At the very least, the GLPs give it a guideline to kind of getting everyone on the same page and conducting studies in a similar manner, at least with respect to the quality management aspects. A couple other compliance notes. So, that was all focused on GLPs because that was kind of the biggie there. But of course, we went through, all the other mentioned regs and guidances. Some of that's not directly mentioned. Excuse me for a sec. But we thought important is also 21 Part 11, electronic records. I'd love to actually hear more about this from FDA, colleagues on this, but certainly I heard earlier, audit trails, right, on analytical data acquisition is key. That's a key part of 21 Part 11 that's even highlighted over there, timestamped audit trails, right? Other systems, for maintaining records and things like that, fully compliant electronic systems can be really expensive. So, our approach there in cases where it's like, wow, we just aren't going to shell out the cash for a you know, a $500,000 limb system. You know, go to paper. It's not fun to deal with, but it's really, paper is fully compliant. So there's really no argument against it there from that aspect. And a couple other guidances. Okay, I lied a little bit. I do want to give my opinion on the analytical guidances. I don't think, you know, either of the cited guidances, which is ICH-Q2R1 or the FDA's bioanalytical method validation guidance, neither one's a perfect fit, you know, for IVRT or IVPT samples. You know, I have my kind of opinions on them here. You know, my opinion is you can use that bioanalytical method validation guidance certainly, again, as a good framework for rigorous methods that can support the sample analysis from both IVRT and IVPT studies. Certainly not every aspect of the that guidance is applicable, especially when you're talking about IVRT type matrices. But again, it's the framework that I think that it provides. That's, you know, kind of the good part there. So that was our assessment, right? So we looked at the guidance, we looked at all the guidances cited in the guidance. We need to figure out where are we kind of missing, where are gaps in our system right now, and how are we going to fill those gaps. Ultimately, it came down to really a lot of you know, kind of paperwork infrastructure. SOPs obviously were a huge thing. Yeah, I mentioned earlier we had kind of just gotten started on writing US specific SOPs, but that was a key thing. Lab ops and quality assurance SOPs, and then also equipment qualification. That was something where, you know, again, the rigor applied to these studies is very high. And so we felt we needed to, again, be very rigorous in our calibration and qualification programs for all this lab kit. So as an example, there's a kind of list of some of the key equipment that at the time that we were using in the lab, and you know, a picture of one of our instruments over there on the right, LCMS instrument. So that was kind of, you know, the list we were starting with. Here's the areas that we need work on, right? And then how are we going to manage this project? Again, I mentioned, you know, we have lots of project managers, so let's plan this out and see how long it's going to take us to do, and you know, when we think we'll be done and how long will each activity take. So we even made our own kind of internal Gantt chart here and to follow that. So for instance, you know, just to write a water bath SOP and get it reviewed and issued, qualified water baths, all those things planned out, when we do them, how long each step would take, just so we could provide, you know, our upper management with an idea of like, you know, just this is going to take a little while to do right, and to get right the first time, hopefully. Of course, I mentioned this before. It's not fast process, nor is it cheap. I mean, these studies, these in vitro BE studies are less expensive than clinical trials, absolutely, but they're not cheap studies. You know, it takes a lot of work to get this system compliant, and then also to maintain that system. So we estimated an awful lot of hours and thought we'd be done in about, you know, 5 or 6 months. We were a little optimistic on that part. But where are we in time? So it's 2017. It's kind of the August-October range in 2017. We really did that full assessment on the guidance, checked our gaps, and planned it all out. September 2017, a key IVRT paper came out. Some may recognize that there, which really is extremely helpful for the conduct of IVRT studies. And then the rest of 2017. So we're deep in progress in November and December on SOP writing and qualification. Some of the qualification, you know, we decided to outsource to third-party vendors. For instance, some of our cold storage units for mapping and things like that was really, even though it, you know, cost us money, we decided it was actually better to get, again, a professional to do that kind of work rather than potentially not doing it the best possible way. So 2018, February, we're a little bit behind schedule. We were a little bit, you know, underestimated how long all this would take. But one key thing that we wanted to do all along was make sure not only did we think we were doing okay and filling these gaps, but make sure that somebody coming in from the outside would do that. So we hired a third-party auditor to come in and take a look at what we put together so far. I think that's a really good recommendation for anyone who is starting this type of work. If you haven't been audited in the past by, you know, your sponsors or anything like that, hire someone to come in and take a look at your systems, and they will definitely find the gaps for you if you haven't found them yet. lucky for us, we had done a fairly good job of identifying our own gaps there. And so really the key things that auditor found were items that we just hadn't quite finished up yet. And then what's happening the rest of 2018? So in April, actually just a couple months later, we have all our SOPs written and approved. All our systems are qualified, and we're actually starting the method development phase of a bioequivalence study. So we are basically on track, and we're doing well there. Just one little wrinkle. It's not really relevant to this conference, but it's relevant to our company and companies like ours. October 2018, the EMA came out with their own draft guidance on topical products. Not identical to the draft Acyclovir guidance, so that's its own set of challenges. Is trying to make things potentially meet both guidances at the same time. I will note that the EMA guidance doesn't really mention a whole lot about quality systems, so that is one interesting difference there. And then where are we, like, people-wise? It's about 2 years later. We're at 27 people in the U.S., so we're getting a staff that can really kind of support these kind of studies. And again, we're well over 100 people at our combined sites in the U.S. and the U.K. Getting close to the end here, so 2019. So we actually had our data included as part of submission to the FDA under this draft Acyclovir guidance. And at the end of 2019, we're up to 41 people here. So 2020, I'm just putting up this email I put up here earlier, right? Which is turns out was not wrong. Said that the FDA would probably come take a visit, and in January 2020, we did get a surprising knock on the door from a couple of auditors from the FDA that were there to review the materials related to the study that we conducted, and that was submitted under the in vitro bioequivalence guidance. What happened? So this is this is extremely redacted here, but the take-home message here is that the auditors were on site for 5 days at our site in the US. Actually, later on, they sent auditors to our site in the UK as well for a week, so they were very thorough. But the end-all be-all was that they did inspect us very thoroughly, and there was no 483, nor were there any significant discussion items. So we felt that went pretty well, and that certainly our systems weren't and aren't, no one's ever will be perfect, but they certainly were well on their way to being working well with this type of submission. So just to kind of summarize here, yeah, I mean, doing these studies is a little bit of a risk. You just have to consider if that's a right thing for you. How much it's going to cost versus how much you could potentially make on it. Are you willing to go through some regulatory inspections? If you decide to do this work, I think you need to do it right the first time. That's not just for yourself, but for the how these products are ultimately going to be used and for safety reasons. And then if you're going to do this, assess the guidance, pay attention to context. Assess yourself, find your gaps, plan it out, make sure you have the right people to do this work, and really couldn't recommend highly enough auditing yourself. Acknowledgements, there's far too many people here for me to really fully acknowledge, but here's some kind of snapshots throughout the years from our US crew, going all the way back to employee count of 7 in December of 2016. And then also, of course, I have to give a shout out to the founders of MedFarm, Andy Muddle and Mark Brown down there in the bottom left. Without them, none of this would have happened. So also thank you all for your attention, and I'm happy to hopefully participate in the panel if my internet stays up. So thank you all very much.